All right. Hello, everyone. My name is Sarah McNulty. I run the Skype Scientist program, and today we are going to be talking all about the artificial intelligence of drug discovery. So how we can use computers and artificial intelligence to help us find new drugs. Today, we are joined by our scientist. Could you say your name for me?
Rocío, Mercado.
Rocío. Okay, awesome. Thank you so much for joining us.
Yeah, no, thanks for having me. Is that a chameleon, by the way, in your background?
Yeah.
Gosh.
He just hangs out back there all day. Awesome. So everybody, thank you so much for coming. Thank you for joining us. Rocío is going to be telling us a little bit about, who she is, what she does, and why she likes it. And from there, this whole conversation is about your curiosity. So if you have questions, either while we're talking or after, please submit them to the Q&A. We will try to get to as many of your questions as we possibly can today. There are no silly questions. Bring it. So thank you again for joining us. Do you want to tell us a little bit about who you are, what you do, why you like it?
I actually made some slides on exactly these three topics. So can you see my slides just now?
They look great.
Okay, perfect. Let me just make it smaller. Oh no, doesn't work. Okay, great. Yeah, so I made some slides on, first of all, my research, very briefly, then a little bit about my career trajectory, and finally a few slides on what it is that I like about my job. So I'll start by talking about my research and the motivation for my research. So first of all, the main motivation is that the drug development process is very long, very challenging, and very expensive. So typically you see it illustrated in some kind of schematic that looks like this, where you have the drug development process divided into 4 different phases. So drug discovery, the initial drug discovery phase on the left, followed by preclinical trials, which is then followed by clinical trials, which is actual trials in humans. And then that all leads to the eventual approval of a single new drug, which goes on and makes it to the different markets. So let me show my little pointer here. Okay. The way the drug development process generally works is you start with, billions and billions of compounds on the left here, which we can computationally screen in order to identify promising drug candidates. And every single step of the way, we're kind of narrowing down the space of viable candidates, let's say. But there's a few issues with this current process. And the first is that it typically requires over a decade to bring a single new drug to the market. So this is going from the initial drug discovery phase here on the left through regulatory approval. The second challenge is that there's generally very high failure rates associated with the types of molecules that do go on to make it to clinical trials, which are extremely expensive. And generally they just fail because they are not efficacious. So they don't actually have any effect for curing the disease, which as you might have thought from previous studies in the preclinical phase. So this all leads to a process which is extremely expensive. And it of course depends on the type of disease, the type of modality, how many patients suffer from the disease and so on. But it's been estimated for some drugs that the development process has cost over $2.5 billion. So this is really, really expensive. And it would be great if we could get that down, because that means we could potentially develop drugs for even more targets, even more diseases, and so on. So we could study a lot more things if this was all cheaper and shorter. And one of the aims of my research is to investigate how we can use artificial intelligence methods to make this process shorter, less challenging, and less expensive. So you can actually use AI, so artificial intelligence, in all 3 of these different phases. But my research focuses on using it for the initial drug discovery phase, where we have lots and lots of data and lots of compounds. And so these methods are really promising. And before explaining the types of models that I work with, I want to just briefly show kind of what it is that we're working with here. So this is a schematic of an artificial neural network, which is a biologically inspired computational model, and it's typically illustrated something that looks a little bit like this. So you have this kind of network in the middle, which is the neural network, and it takes some kind of input data and returns to you some output data. So this could be input, the input could be molecules and the output could be properties for those molecules, for example. But what makes these models really, really great is that certain classes of them can, in theory, approximate any kind of mathematical function. So they're really powerful models that we can use to learn various things. And so I use neural networks in some of my research areas. One of these, which I've depicted here, is molecular design. And here, the idea of molecular design using neural networks is that we have a model for our neural network on a computer, which we code up, and we feed into our model some kind of desired properties. So the properties we want for the molecules to have are potential drug candidates. And we let the model do the optimization and design for us the molecules which it believes best satisfy the set of input criteria. So these type of desired properties could be things like target activity, low toxicity, good solubility, and so on. And the model can generate the different molecules in autoregressive fashion. And for instance, here it's just generated aspirin. So in practice, in reality, we wouldn't care about generating aspirin again, but this is just for an example here. And we could use this for a wide range of different problems where we have different sets of desired properties for our drug candidates to have. So this is all computational, even though I showed some of these things happening like on the sides of the computer. It's all computational drug design. Then the idea once we generate these molecules is that they can go on to be synthesized and tested in a lab. So you might want to test it either in cells or they may also be tested in animals. If this is successful, the molecules can then go on to also be tested in humans. And this is where you really need to show that the drug is doing what you claim it does. And then following, very stringent criteria, if your drug candidate does satisfy all these very specific criteria for that a new drug should satisfy, then it can go on to be approved and sold to different markets, in which case you need to manufacture it and so on. So again, my research is in the computational drug design, where our goal is to design better molecules early on so that we can increase the likelihood of success in later on experimental studies, which can be very, very expensive. So that's it in terms of kind of summarizing one of my main research areas. Now I want to switch and just touch on my scientific trajectory briefly. So here I show a little family in Mexico, and that's meant to represent kind of my parents and how they immigrated to the United States around 1992 when I was born. So I was born and grew up in Los Angeles. Then I got started in my scientific career when I went to study chemistry at Caltech. So this was between 2009 and 2013. I got my bachelor's in chemistry. So put this little animation. Then after this, I moved to Northern California, where I went on to do my PhD in computational chemistry at UC Berkeley. So I have another, yeah. But throughout my PhD, I was doing a lot of traveling. So I also traveled abroad to Switzerland quite a bit, where my boss from Berkeley was starting up a new lab at EPFL. So I was a visiting PhD student here. After my PhD, I traveled, I moved to Sweden, where I lived for a few years. And here I did an industrial postdoc at AstraZeneca. And it's actually where I got started on artificial intelligence for drug design. Following this, I moved back to the US, where I'm currently now doing my second postdoc here at MIT and continuing work along the same lines. So what do I like about my job? I guess the main thing that's to me the most important is the possibility to help people with my research. So building models that can help other researchers find therapies for diseases, models that can help us repurpose known therapies, models which can help us develop a better understanding of certain diseases, and of course, publishing these tools open source and freely so that others may also use them in their research. And in this way, kind of maximize the impact of one's research. So this is the main thing that I like. The second is that I get to work with a very, very broad range of amazing people. So I've worked with really great colleagues and students over the years, and this is just a subset of those people. So I actually couldn't find any pictures of my current research group, but they are also really great. I just couldn't find any pictures to show them, but really the people that you get to work with in this field are really incredible and make it a really satisfying job, besides the fact that you can actually have an impact with your research. And then of course, like the traveling and getting to live in a very broad range of beautiful places. So these are pictures from Switzerland. So I'm standing right there and ruining my friend's almost perfect picture of this Swiss landscape. So this is also Switzerland here, where I was working when I was traveling back and forth as a visiting PhD student. And then of course, Boston, and here's a picture of Gothenburg and Sweden. So all of these things make it a very rewarding job. And I'll be happy to answer any questions that the audience may have, whether it's related to my research or career advice or chemistry, whatever it is. So yeah, thank you.
Amazing. Thank you so much. Yeah, I think I'm so glad that you gave us a map of everywhere you've been, because I think a lot of folks don't realize how much you travel as a scientist, both for jobs and just for work. So that was awesome. Thank you so much. Okay, we have a bunch of questions from Mrs. Sharp's 802 class from Brooklyn. So here we go. Number one is, how much data can a molecule store?
That's actually an interesting question that I don't think is how they intended that question, but you can actually store information in things like DNA. So this is a big area of research using this to store the large amounts of data which we are generating. However, for like the types of models that I work with, if maybe what the question was getting at is like how many molecules we're typically working with. We're typically working with like billions of both hypothetical and actually synthesizable molecules to train our different models. So it really depends, because there's also cases where you have very few examples of molecules which are actually, active against a specific protein target and might be, good drug candidates. So sometimes you're working with very few data points, but in general, to train neural networks, the more data you can have, the better.
Very cool. The next question is, how long did it take to get a PhD in chemistry?
So it took me 5 years, which I think is, I think it's quite normal for in the US. If you go to Europe, typically the PhD is a bit shorter because you do a master's first. But I mean, it takes people a very wide range of times. It took me 5 years.
Yeah, awesome. How do you like working at MIT?
I love working at MIT. I think I love my colleagues. The department is also great. There's so many different seminars, great facilities. I have a great supervisor, so I really enjoy it.
And Boston's such a fun town to do science.
Yes,
There's
exactly.
just scientists coming out of the woodwork. That's always really fun. All right, what is your favorite thing that you've personally discovered in molecules? Like, what is this discovery you were like really excited about?
Well, the thing is, I'm not using my models necessarily for specific applications themselves at the moment. I'm more building the models that I hope other people will use in order to discover promising molecules. So doing the method development, but that's what's been really exciting for me, is kind of building this model, which we call the graph invents. It uses molecular graphs and publishing that and seeing like people actually using it and asking questions. Like even if they're having trouble running the code, it's still like very satisfying because they're actually using it enough to find problems. So this has been my most, I guess, satisfying thing.
Very cool. We got a question from Sam. What languages do you program in?
Python for almost everything. I have used other programming languages, especially during my PhD, but now there's so many nice libraries for different like machine learning and AI tools in Python that's kind of what most people in the field use.
Yeah. Skype a Scientist matching algorithm, also written in Python. So you can do everything from drug discovery to classroom scientist matching. So we can do it all with Python. I didn't code that. I don't know. I can't code myself out of a paper bag, but a person who works at AstraZeneca, actually, the things we keep lining up is the person who wrote that.
Yeah, that's right. Okay.
Yeah. All right. The next question is from Trenton. What is the minimum length of time it could take to make a drug? Like everything goes great and it's smoothly flowing about what's the fastest?
That's a very interesting, like, hypothetical question. I have heard really interesting stories about certain like drugs, for example, Tagrisso, which is used to treat lung cancer, I believe. And I think this was also developed at AstraZeneca. And this was also a really quick development timeline. I want to say 5 to 7 years, something like that, which was quite quick, actually, for a drug development process. But I don't know what is the quickest. That's a good question, something I can look into.
Cool. The next question is from the Cincinnati Zoo. Cool that the Cincinnati Zoo is here. Hey, everybody. What are the up and coming research priorities that you see in your field?
And for me, so one of the areas which I'm actively working on now is synthesizability, constrained molecular generation, which is kind of a mouthful, but the idea here is that we can generate lots and lots of molecules. We can generate billions of molecules on a computer, but there's no requirement that the molecules that we're generating are actually synthetically accessible. So there's no guarantee you can actually make them in the lab, and that's kind of a problem. So being able to use like reaction information, which we also already have, and incorporating it into our models in order to allow us to guide the generation towards molecules which we actually can make in the lab. I think this is an area of research which I'm very excited about. There's a lot of excitement as well for things like computer-aided synthesis planning, so using this reaction information to also predict the synthetic pathways to other molecules, so which you generate computationally. So you actually have a recipe for how to make those molecules. There's a lot of excitement about like 3D molecular generation. So because a lot of the data structures which we work with is just 2D representations, which means we don't have actually the coordinates. And in reality, molecules, they're rapidly interconverting conformers, right? So this could be a potential limitation of the types of models we have now, and there's a lot of work aiming to incorporate 3D information into other models. So these are some of the big directions which people are excited about. I'm excited about them as well. Being able to also develop models which can learn from very few examples as well. So oftentimes in drug discovery, we only know a few examples, a few molecules which actually bind to a specific protein. And we need to be able to extract as much information as we can from those few examples in order to design other better drug candidates. So low data learning, that's also a very hot topic.
Very cool. So like of the molecules that the computer program spits out, let's say, you know, you're trying to reduce pain in a way that isn't addictive, for example, and you come up with 100 possible options. How many of those 100 can actually be synthesized? And does it depend on the project or like how does that kind of translate?
It depends on a few different things, because there's also like a big current like shift towards automation. So if you can actually, if all of those molecules can be synthesized using a very small set of reactions, which you have the capacity to automate, then it might not be a problem at all to generate, to synthesize all of those molecules. But if you start to require more complicated synthetic pathways for generating certain molecules, then that's going to become both more expensive in terms of time and in terms of money. So I would say on the order of thousands for sure, because sometimes you have these molecules already in libraries, which you can screen. Maybe you hadn't already screened them for a particular application. And then for synthesizing new ones, maybe a couple hundred is feasible. I hope I'm not saying like completely crazy orders of magnitude, but yeah.
Cool. So like, okay, that's cool. The next question is from Sam again. What types of animals are commonly used for testing? And are some types of animals better suited for research questions than others?
Yes. So the whole animal testing thing is quite complicated. So it tends to be that for like the easiest, you first would maybe want to do experiments in cells, and then move up in terms of complexity. So you can do experiments in rats and mice. I know they also do experiments sometimes in like dogs or pigs, but this is models which are typically used after the fact. So after you have done already experiments and you're getting ready to move on to maybe clinical trials, you might go on to these higher species, but you wouldn't be trying to do experiments which is going to kill the animal. By this point you have an idea that the molecule is going to be safe, and maybe you want to study something to do with the dynamics or kinetics of how this molecule behaves in the body, let's say, of the different animals, and it can help you determine the dose that you're going to use when you go on to actual trials in humans.
Awesome, thanks. The next question is from Dave. Can this concept of a neural network behave similarly to the ones used on protein folding? And for a little background for everybody, you may have heard of programs like called Fold at Home. Like you can use these in your classroom. You can use them at home, anywhere. It's basically a program that takes proteins. So proteins, as a background, start as really, really long strings. And in the body, they don't look like really long strings. They look like tangles of string. So instead of being long and skinny, they're like a little ball of spaghetti. And how they go from long string to ball of spaghetti is sometimes a mystery to scientists. They just kind of make it work, and you can kind of predict how that happens. And so one of the things that many scientists are trying to figure out is how they come into being the folded, crumpled version that they end up actually looking like in your body. And so people absolutely are using computers for that as well. So is that similar to the stuff that you're doing?
With the question related, sorry, I forgot.
I'll read the question again. Can the concept of a neural network behave similarly to the ones used to work on protein folding?
Yes, so there are people using like neural networks to predict the folded structures of proteins. So you may have heard of also like AlphaFold and AlphaFold 2 recently. So they use neural networks in order to help predict the folded structures of proteins. Yeah, that's another big area of research, actually.
Yeah, very cool. Next question, how much of your day is spent coding?
It depends. It depends on the stage of the project, right? So earlier on in a project, you might be doing a lot more coding, and maybe it looks like most of your day is actually spent coding or reading papers. But then later on in the project, you might be done with your code, and you might actually be using it to run different experiments on the computer, and then analyzing your data. So earlier on in the project, it could be like most of your day, at least for me.
Yeah. All right, the next question is really interesting. It's from Rob. So can AI help identify drugs that work in women? So I'm going to give a little bit of a context to this question. I think there's this problem in medicine broadly that a lot of the drugs that we use were originally tested in like white men or white people in general. And so different groups of people don't have as high efficacy with the same drugs. Like, can you use AI to better figure out what's going to work in different groups of people?
Yeah, so this is also a huge area of research, right? So like precision medicine, or trying to understand how different populations are going to respond maybe differently, or how differently to maybe the same treatment. And you can certainly use like artificial intelligence in order to predict this. So either working from data on the genome, so do certain populations have different genes that others do not, and that makes it so that they're going to respond differently to a specific treatment. Also on the proteome or the metabolome. So there's work on all sorts of these areas. So using AI in order to learn from all this data which we're generating, and which we have both on the individual level and on the population level, and using that to predict how differently populations will respond to treatment.
Very cool. And a proteome is like your genome is the DNA. So that's all your DNA that you got in your body on the whole. And then your proteome is the proteins that the DNA coded for that are like functional, actively floating around and doing the work of being alive, basically. So it's like you could have stuff in your genome that never actually gets turned into a protein for whatever reason. So having a proteome is really, really useful. So a lot of things end in OME, OME, and that's like all the stuff from the situation that you were studying. So proteome, protein, all of it. Genome, genes, all of it. So when you hear ohms, that's what we're talking about. And we stick ohms on everything in science. It's all over the place. Metabolome, more than that. Metabolome, metabolic. Next question is from Johan. How does working for a university compare to working for a company?
Yeah, there's differences for sure. I think there is advantages and advantages to either one. So in a university, you for sure have a lot more freedom to explore the problems that you're interested. And then depending on the company that you're working for, you might also have better resources actually with which to tackle these different problems. So it depends on the university, depends on the company and the amount of funding in either case. At companies, you also often have the advantage of having lots of nice internal data, which you can use. So data which was generated from projects, like let's say the company was working towards developing all sorts of different medicines, and through these different projects, they've generated all this data, which is not public. So the company owns it and they can use it to get a competitive advantage over other companies. So this could be an advantage of also being a company, but in general, in academia, you have a lot more freedom. You can also form collaborations with all sorts of other companies, perhaps to get access to this data. So there is differences, but advantages to both. Hope that answered the question.
Have you found a difference in like your work-life balance between working at like institutions like MIT are like at least I've heard, are very, very intense, and you're working like kind of all the time? And then at AstraZeneca, maybe you were working 9 to 5 and then going home. Have you experienced that personally?
Yeah, that depends a bit on the individual, right? And the types of boundaries you set for yourself. I definitely feel a lot more pressure, like especially being at MIT. Whereas at AstraZeneca, it's also like, it depends a bit also on the age range of your colleagues, right? Because now at MIT, it's a bit like working in a group with other postdocs and PhD students, and the crowd is much younger. Whereas at a company, you tend to have also a lot of older people who have a lot of family commitments. And so they tend to stick a lot better to the 9 to 5 schedule. So yeah, there is differences, but you can also control for that. There's also people who work nonstop in
both
in industry. So yeah, you can find both types of people everywhere.
Cool. Next question is from Piper. How long have you been coding in Python?
That's, I guess, since the beginning of my PhD. So I did my bachelor's in experimental, like synthetic and organic chemistry, and I actually couldn't really program by the end of that, but I decided for my PhD, I wanted to go into computational chemistry. So I had to learn all that in my first year of my PhD. So whenever that was, since 2013, I guess, I've been programming.
Awesome. All right, the next question is from Trenton. What is your least favorite part of your job and most favorite part of your job?
Least favorite part of my job. I mean, the least favorite part of my job. I really, really love my job. I guess like not having enough time. There's just way more ideas than you can come up with than what you can actually study, and not having the time to explore all of these. So this is why it's like becomes a good idea. At some point you want to start your own research group and you want to hire like students and other researchers to also explore these ideas with you. So that for me is hard. Also like choosing what to work for. In academia, at least it can take up a lot of time to apply for things like funding, research funding for your ideas. And it can be a bit random sometimes, like who gets the funding. So you might think you wrote a really amazing proposal, but in the end it does not get funding, but you try again. And that's just the current working model for that. So maybe that would be my least favorite thing. But
grant writing is probably, it's up there with the worst part of my job too. All right, the next question is from class 802. What websites could students use to begin learning about coding or biology?
Oh, that's a good question. Websites for learning about coding. I think there's a lot of good online resources. Like, I don't know what's the grade level of this course, so I don't know if it's suitable for like elementary school kids and whatnot. But I know that there's websites like DataCamp and Coursera, which lots of people use to like learn the fundamentals, and also you can also get into much like higher level content there as well. For biology, I don't know if I have a specific website, but there's excellent videos on YouTube, right? You can find online courses that sometimes, especially now during COVID, a lot of professors have recorded their lectures and made them available. So if you search for like an introductory biology course, you can find this on YouTube for sure. And it's a very good qualities. So yeah, I would suggest YouTube for.
Yeah, I don't have one go-to biology website, but there's so much content online that it's kind of everywhere, and YouTube is great for it. And having like noting that the account that you're looking at is like a university account, that's often really helpful. Yeah. All right, the next question is from Sam. Can you show us what your programming environment looks like, or a project that you're currently working on?
What my programming environment looks like. I mean, I can describe it. I don't know if it feels too risky to share like my screen and like everything that could be wrong in my current code. But I use like Visual Studio Code for development. I guess most of my day is doing like remote development. So on a remote machine, as I said, in Python. A project that I'm currently working on. So I've recently published some work with Wen Hao, who's a PhD student in my current group, on synthesizability constrained molecular generation. So this is something which we're still actively working on. So we published a model called SynNet. So Syn for synthesizable and Net for network. And that's still an area which we're actively investigating how to improve the predictive performance of these different models, as well as the reaction scope. So that's maybe a bit too detailed, but yeah.
Cool. Dave wants to know, have you ever done any research that's not related to chemistry?
I mean, that's, where do you draw the boundary? Because technically a lot of the research I did for my PhD was actually in like, could be called material science, because I was doing computer simulations of nanoporous materials. But if you ask me, as a very biased like I think everything is chemistry, right? So how are you? Yeah, it would be, I guess short answer is no, I have not done something which I would not consider chemistry.
Cool. Antonio wants to know, what's the favorite place you've ever traveled to?
That's a hard question. Yeah, I don't know if I can answer that question. There's like really good things about everywhere. I've left when I was working in Sweden. So I felt that, yeah, I had a really good work-life balance, and it's really easy to get out into nature from anywhere that you live. So even going just for like a 5-10 minute walk, and you can be out like in the middle of the woods. And I thought that was really nice. But I don't know if I have a single favorite place. Maybe that's just my favorite place I've worked so far.
Cool. Sounds good. Next question is, what job would you do if you weren't doing your current job?
There are these really hard questions.
I don't know what I would be doing if I wasn't doing this.
Yeah, no, I don't. Maybe I could go like, if I was forbidden to do any science at all, maybe I would go to like culinary school and try to become a better cook and do this. But that seems also like a very stressful job. So
both like high intensity jobs for sure, being a scientist at MIT and being a chef. Both require protocols, both require that you pay attention to what you're doing. I bet those skills would transfer. I bet you'd be a great chef.
Oh, thank you.
Yeah, sure. All right, the next question is from Nick. Who are the people using the models you build?
So there's a range of different people. So there's different groups at like other research institutes, universities. I would hope that there's also groups at specific companies, although this I don't know for a fact. Most of the people I talk to who are using my tools are like at universities or research institutes where they're working with like publicly available data. So other researchers, like other PhDs and so on.
Cool. There are a couple questions in here that are asking similar things. Basically, like, when did you first think I want to be a scientist? Like, what inspired you to be a scientist, and when did you first think that that was something you might want to do?
I think in the 6th grade, when I mean, your teachers, right? Like when you're doing really well in your science courses, I had a very supportive science teacher, and he was like, "Yeah, you would be a really great scientist. You should study this more." So then, like, that's the seed is planted, and you go on to like high school, and you choose your like electives in high school based on this. I applied to college, knowing that I wanted to do something in the sciences. So I guess for sure, I didn't decide I would study chemistry until I was a freshman in college, but yeah, I guess the seed was always kind of there since maybe the 6th grade.
Very cool. All right, the next question is again from Mrs. Sharp's class. This is Zach asking, what is your favorite part of working specifically at MIT?
I really like my colleagues. I think I've already said that. I think the group I work in now is amazing. I really like my supervisor. I think it's just a really like nurturing environment for like, you know, coming up with new ideas and pursuing the ideas that I'm really interested in pursuing right now. So that would be my favorite thing. My colleagues, I guess.
Yeah, that's great. So how many people are in the working group that you're currently in?
Maybe between 10 and 20. Someone else just asked me this recently, and I counted and I forgot. I think maybe
somewhere between 10
like that.
Sounds good.
Yeah.
And labs can have like all different sizes. Like the lab that I worked for getting my PhD, we had like 5 people in the lab, including the PI. And that fluctuated between like, let's see, 3 at the least and 7 at the most. So it's like, there are all different sizes of labs. Some labs have like 50 people working for them. My buddy got his PhD working in a gigantic lab at the University of Pennsylvania. So it's widely variable. All right, this is a hard question that I don't know how you're gonna answer, but here we go. Coming in from Lucas, which animal is the easiest to test on?
That's very hard, especially because I'm not an animal expert.
Yeah.
So I don't know if I can answer this question.
It depends on the question that you're asking. Like sometimes it really, you start with your question and then you pick the animal that's the right animal to ask that question. So there's no like broad answer for this is the easiest of the animals. Although I will say working with cells is easier than working with animals by a lot. I've worked with animals, I've worked with cells. Cells are just so much easier. You still have to feed them, you still have to take care of them. It's kind of like having a pet plant, except a little bit more challenging than that. But it's somewhere between having a plant and having a cat, having some cells.
I guess another interesting area, like application of AI in drug discovery, is also the development of these computational animal models that you can use to predict some of the properties that you would be measuring in the animals before you actually test an animal. So to minimize the amount of animals which you actually need to use for testing. So this is another cool application of AI in the area.
Cool. I'm going to read through really, there's like a long list of questions. I want to make sure that we got to them all. So to give me just a minute. All right, how much data can a molecule store? We got that one. We got that one. Okay. Okay, here we go. This is from Rob. Can you also do drug-drug interactions via AI to know if maybe like this drug works for this thing, but if this person is also taking this drug for this other thing, they're gonna not work together?
Yeah, this is another like big area of research in like AI applications to drug discovery. So there's people who can build like knowledge graphs from biomedical data, and you try to find kind of the different nodes in your graphs might be different molecules, different protein targets, maybe different diseases if you want to label it in some way, and you want to find links between the molecules and the different protein targets and the different diseases. And sometimes you can also find links between other molecules. So using things like knowledge graphs can help you learn something about potential drug-drug interaction. So there's a lot of work I've also seen from Regina Barcelay's group at MIT, where they built some models for predicting if 2 drugs are going to have a kind of synergistic effect on each other, or if they're going to hurt each other's performance. So there's a lot of work in this area. Yes.
Very cool. My buddy, who also lives in Philadelphia and got his PhD where I got my PhD, is working on, I don't know if it's called molecular dynamics or if he works on molecular dynamics and also does this thing. So you might know the answer to this more than I do. But when he presents on his work, he shows these videos that basically looks like 3D models of molecules and 3D molecules of either like the surface of a cell, like super, super blown up. So you see every one of those little lipids in the cell surface, in the lipid bilayer you may have learned in class, everybody at home. And like, it just looks like they're all just kind of like jumbling all over the place, because that's kind of what happens in our cells, because everything is so small. And it's like, they just sort of like bump into each other. And then eventually they bind, like the, whatever is necessary for the binding to happen happens. And so he'll take like 3 different versions of a drug that are slightly tweaked and look to see which one binds the hardest, binds the fastest. And those videos are both funny to watch, because it's funny to think of a drug like bumbling through its day, and also just very, very cool. So is that molecular dynamics, or is that something?
Yeah, that's molecular dynamics. And you can use AI like to develop like force fields, which you can use to actually like run your molecular dynamic simulations to predict the energies and so on. So yeah, but it is really funny, like to watch the simulations. But that's what molecules really look like. That's what they're doing.
Yeah, yeah. You think of things like when you watch videos on YouTube, maybe like you think of everything moving smoothly, and just like the artistic renderings of cells are often so like beautiful and smooth, and then you watch like the actual best description of reality, and it's much more bumbling. It's very funny. Highly recommend everybody just like take a look at a molecular dynamic video after this talk. Okay, so we try to ask everybody the same 2 questions at the end of every session. We're almost at 45 minutes here. The first question for you is, if you had everyone's attention in the whole world and you could tell them one thing about your area of expertise, what would it be?
Let me think about this for a second.
Sure.
I guess the main thing I would say is that I really do believe that, I mean, we have so much data which we're generating these days, and so much data which we have generated over the past few decades, that I really think the time is right now to be kind of using these AI models in order to both predict properties about molecules, predict new reactions, to design molecules. I think this is kind of like the best time to be working in this field. And I guess to anyone who is listening, I mean, there is so many also, it's such a hot field. There's so many jobs in the area as well, that I guess I would encourage lots of people to look into this if they think it's something they're interested in. To me, it's such a exciting field, and it holds a lot of promise for how we can actually like accelerate the drug development process using these tools. It remains to be shown. So it is not like guaranteed that these kind of methods are going to actually do anything to speed up the drug development process. But I think that they're very promising. And that's something which I think people should look out for and see. This could all lead to, if we can do this, if we can use AI to, more quickly develop drugs, then that's going to allow us to study, a broader range of diseases, a lot more molecules, possibly lower the cost also of drugs to patients, and yeah, it could really reduce the amount of suffering in the world, right? So it's a research area which I'm very excited about, and I hope others are as well. So there continues to be funding in this area.
Great. Sounds good. All right. Your next question is as hard. Here we go. You still have everybody's attention in the world. You can tell them one thing about literally anything. It can be about science. It could be about the cows you met in Switzerland. It can be about whatever you want. What do you tell them?
I would tell them to visit Sweden. I think Sweden is a wonderful country, and I think it's a bit underrated, especially maybe for folks coming from the US. So I would encourage people to go, and yeah, I really love it. So
that's great. Sounds good. Sounds good. All right. Okay. Do you have anything else you want to share with us? Do you have places you want us to go? Things do you want us to follow you on social media or anything?
As you like, you are free to follow who you like. I'm not going to tell you to follow me and then be disappointed by my lack of content. Like, I don't post that much. So no, it was really nice to tell you all about my work and the area in which I work in. And I hope you guys are all like leaving a little bit more excited about this research area as well.
For sure. Well, thank you so much for sharing with us. This was really cool. I learned a lot for sure. I think everybody else did too. Thank you again. And thank you, Kay, for signing. We always appreciate you being here. Everybody else, the next time we meet is going to be in February. We're going to be talking about space-time. So space-time, what the heck does that mean? We're meeting February 23rd. That's all about like how time moves differently in different parts of space. It's super weird. And when you think about it, sometimes it feels like your brain's going to explode. It's so weird, but so cool. We're going to be talking about a scientist who's an astrophysicist who studies gravitational waves and black holes and all sorts of wild space stuff. So come join for that. When we don't know the answer, sometimes in biology sessions, I can fill in gaps. And with that one, I definitely don't know anything. So I'll be learning right along with you for space-time. So we'll see you then. Thank you again, everyone, for coming. Thank you for sharing your knowledge with us. This was really awesome. We'll see you again next time. Bye.